So after this great talk, so we're gonna move now to the young scientist talks. So the first talk will be from Priscilla Masamba from University of Zululand in South Africa. Hi, Priscilla. So I'd like to ask you to stick to the 15-minute time for your presentation and please start sharing your presentation with us.
Okay, can you see my slides and can you hear me?
Yes, we do.
Okay, thank you very much. Good day, everyone. My name is Priscilla Masamba, and I'll be presenting on determination of the 3D structure of Schistosoma mansoni universal stress G4LZI3 protein by X-ray crystallography. Schistosomiasis, which is commonly termed bilharzia, is one of the most potent water-based diseases in both tropical and subtropical regions of the world from a global health perspective. It affects more than 252 million people, with 90% of all infections occurring in sub-Saharan Africa and taking the lives of 300,000 people per annum.
Every month we are doing this. And
according to Adenauer and co-workers in 2015, countries which are most affected in SSA include Nigeria, the Democratic Republic of the Congo, Tanzania, Mozambique, as well as Ghana. And according to Magaisa and co-workers in 2015, over 5.5 million people are estimated to be infected with the disease in South Africa alone. Schistosomiasis is mainly caused by 3 species, which are known as Schistosoma mansoni, Hematobium, and Japonicum, while Macongia and Intercalatum are less prevalent species. The life cycle of the worm begins when an infected host defecates into open water sources, where merasidia hatch from the eggs under the most favorable conditions and develop into sporocysts, which infect an intermediate snail host. The infective part of the cycle is then released back into the watery environment in the form of Sicariae, which develop in Schistosomulae when they infect a human host, which in turn develop into adult pairs that mate, lay eggs, and then begin the cycle all over again. The signs and symptoms of the disease are more of a result of, they begin with common flu-like symptoms, starting with a headache as well as a rash, which mostly affects people who travel to endemic areas for the first time. However, these symptoms may digress into more serious complications such as liver cirrhosis, periportal fibrosis, malnutrition, as well as anemia, particularly in young children. The manifestation of schistosomiasis is more of a result of eggs which become trapped in host tissue that release proteolytic enzymes that become carcinogenic and thereby predispose the infected to bladder cancer, as well as other opportunistic diseases such as human papillomavirus, female genital schistosomiasis, as well as HIV and AIDS. Ever since the 1970s, Praziquantel has been the most dependent chemotherapeutic drug for treating all schistosome infections, mostly because of its limited side effects, as well as its cost effectiveness. However, there have been concerns about this line of treatment, mainly because it fails to interrupt the transmission cycle, and there have been emerging reports on the emergence of drug-resistant strains. And this has therefore opened the need for the search for alternative treatments. One of the enduring abilities of an organism is its ability to respond to its environment and the stresses that are associated with it. And this may be in the form of high salinity, toxic chemicals, extreme temperatures, as well as drought. Schistosomes possess stress response mechanisms in the form of heat shock proteins, as well as universal stress proteins, which is what this project is based on. Universal stress proteins are a large group of conserved proteins which are found in various organisms that include bacteria, protists, fungi, as well as plants, and they form a large part of their defense system. In bacteria, they are known to function in oxidative stress defense, ion scavenging, motility, adhesion, as well as cell aggregation, and they're classified based on their ability to bind ATP, as well as the presence of various motifs which are thought to play a role in their biomolecular function. In 2016, Masamba and co-workers hypothesized on how the ubiquitous nature of universal stress proteins could be new targets for therapeutic interventions against schistosomiasis. And this was based on a study by Mba and co-workers in 2013, who highlighted or who showed the upregulation and overexpression of universal stress proteins in all the 3 main schistosome forms that affect humans. They prioritized the G4LZI3 universal stress protein as a lead target for future schistosome interventions. However, the biomolecular function of universal stress proteins are not known and are still yet to be elucidated. The aim of this study was to express, purify, and characterize the universal stress G4LZI3 protein for structure determination by X-ray crystallography so as to use the three-dimensional structure for structure-based drug design using natural compounds. The aim of the study was achieved through the following objectives, which will be discussed during this presentation. The gene of interest was first cloned into the multiple cloning site of a PQE30 expression vector using upstream BARMH1 and downstream HIND3 restriction sites. Thereafter, the gene of interest was amplified correctly at 555 base pairs, and this was followed by successful restriction, digest, and ligation into an expression vector. The expression construct was then transformed into M15 cells, where positive transformants were used to screen for the expression profile of the protein. And this showed that the protein was able to express on a 1 mil scale successfully. The gel above shows odd numbered lanes, which represent samples which were uninduced, while the even numbered lanes represent samples which were induced with 0.5 millimolar IPTG. Thereafter, the best expressing clones were then used to set up a midi scale expression of 500 mils and later a large scale expression of 2 liters. This showed that the protein was still able to express at optimal conditions of 0.5 millimolar IPTG at a temperature of 25 degrees. Thereafter, Western blot was then done so as to further confirm the presence of the protein as well as to confirm its size. And this showed nonspecific binding and the protein still expressing at 20.3 kilodaltons. The samples were then subjected to affinity purification, where the protein was purified using an ACTA FPLC purifier system, and the protein was eluted using 400 millimolar imidazole. This showed partial purification of the protein, and the eluted fractions were then subjected to size exclusion chromatography. The resulting chromatogram showed acceptable separation between high molecular weight impurities as well as the protein of interest. And the eluted fractions were then subjected to crystallization trials. Before then, however, the protein was subjected to preliminary biophysical characterization so as to assess protein conformation, and this was first started off using FTIR. The spectrum showed the presence of 3 prominent peaks, where the first peak represents the first amide group, the second peak represents the first peak represents the amide A group, the second peak represents the amide 1 band, and the third peak represents the secondary amide 3 band. Thereafter, DSC was then done so as to determine the thermodynamic stability of the protein. And this showed a large peak extrapolated at 110 degrees centigrade. This showed that the protein is thermodynamically stable at temperatures lower than 110 degrees centigrade and only denatures at temperatures higher than that. Lastly, fluorescence spectroscopy was then done so as to summarize protein characterization. This technique is basically based on the excitation of aromatic amino acid residues such as tryptophan, tyrosine, as well as phenylalanine. And this showed that at an excitation wavelength of 270 nanometers, the maximum fluorescence that was emitted was 344 nanometers. And this showed that the protein was both folded and stable. These results were submitted as a manuscript and have been accepted and recently published in Advanced Pharmaceutical Bulletin. After determining that the protein had not lost its conformation, the protein was then subjected to crystallization trials. This was done using 17 mg per mil of protein in a 1:1 ratio using the Morpheus crystallization screening kit. And this was also done using the vapor diffusion sitting drop method. So the first panel represents the first crystals that were acquired, which were used for microseeding. And the second panel represents the progression of seed growth, while the final crystals represent the crystals that we selected for diffraction. Diffraction data was collected remotely using the Diamond Light Source synchrotron from the UK. And despite the acquisition of various data sets, the best resolution that was acquired was around 3 angstroms. The space group showed that the crystal belonged to the orthorhombic crystal system with 4 molecules estimated in its unit cell. Thereafter, molecular replacement was then used to acquire the initial electron density map. And because of the acquisition of low resolution data sets, extensive manual building as well as correction was done in COOT, while refinement of the model was done using Phoenix Refine. A partial structure of the G4LZI3 model was then acquired, which showed that the model contained 4 alpha helices and 4 beta sheets. One of the objectives of this study, as mentioned earlier, was to use the 3-dimensional structure for structure-based drug design using natural compounds. And this was based on a study by Anandi and co-workers in 2014, who showed that natural polyphenols are able to downregulate universal stress proteins, but this was done in Mycobacterium tuberculosis. So this project then adopted a similar approach by docking 10 polyphenolic compounds onto the G4LZI3 model and selecting the best 5 complexes based on their Z-dope score for further analysis. Molecular dynamics simulations were then done using the GPU version of the AMBER-18 package, and this was run for 100 nanoseconds. The root mean squared deviation showed that all the complexes reached a state of stability and that the initial fluctuation, which is initially shown in the simulation, was due to the polyphenolic compounds positioning themselves within the active site of the protein. Most notable was the curcumin complex, which showed a very sharp decrease in its value at around 60 nanoseconds. And this complex, along with the quercetin complex, exhibited the highest fluctuations of 4.55 and 5.23 angstrom, respectively. The root mean square fluctuation was then done so as to determine the restriction of intrinsic flexibility within the protein once the polyphenols were docked onto the G4LZI3 protein, as well as the rearrangement of amino acid residues. This also showed high fluctuations at the start of the simulation, which then decreased, showing that the polyphenols were able to downregulate the activity of the universal stress protein. Catechin, dalbergin, as well as the curcumin systems represented those which had the lowest average residual fluctuations during the simulation. The radius of gyration basically speaks to how efficiently or how effectively the polyphenols were able to reduce the fluctuation of the C-alpha backbone of the protein. And this showed that for at least the first 50 nanoseconds of the simulation for all of the complexes, there was a high increase in their value, but then this was followed by a decrease in their value for the remainder of the 50 nanoseconds. Once again, the curcumin complex exhibited a very sharp decrease in its value at around 60 nanoseconds, which was in line with its RMSD value, as well as that of quercetin, which also showed a decrease in its value at around 20 to 25 nanoseconds. So this basically showed that the binding of the polyphenols onto the protein was able to basically perturb and downregulate its activity such that the G4LZI3 model or the G4LZI3 protein had lost its compact structure. The complexes were then grouped according to their binding affinities, and this was done using MFGBSA free calculations. From all the interactions shown, catechin and curcumin exhibited the lowest binding affinities. And from this study, we were able to propose that these two compounds should be investigated along with Praziquantel treatment as nutraceuticals, basically because polyphenols are known to scavenge reactive oxygen species, which is a part consequence of Schistosomiasis infection. This is basically a diagram showing how these polyphenols, So this diagram basically shows the change in conformation that curcumin and catechin conducted once bound onto the G4LZI3 protein during the 100 nanosecond simulation. And some of these findings have been published and have been accepted recently in current drug discovery technologies. I'd like to acknowledge the following individuals for their contribution to this project: Prof. Capo, as well as Prof. Sewell as my supervisors, Dr. Brandon Weber, who helped with protein purification as well as setting up the crystallization trials, the University of Johannesburg, where I'm currently doing my postdoc, the University of University of Zululand, where I conducted my PhD studies, as well as the National Research Fund for funding this study. And I would also like to thank you for your attention. Thank you very much.
Thank you, Priscilla.